let's get started. So the outline, I will talk a little bit about what bioimage analysis actually is, some basic concepts and terminology, image data format, some established tools, some practical advice, and then we do the question and answers. So you see the word some there a lot, and the reason is that I cannot really cover all of bioimage analysis now in 30 minutes. So a real course would take weeks, so I just had to pick some things, and I just can hope that it's the right level for you. So, and if not, please excuse, I had to make some choice there. Okay, so bioimage analysis, another word, by the way, that's kind of becoming popular now is bioimage informatics. So the usual workflow of experiment you see here on the screen. So involving microscopy and image analysis and hopefully a publication in the end. And let's say the core business of bioimage analysis would indeed be the image analysis part, but actually to do this job properly, you have to consider all these aspects. And the reason is that very often during the image analysis, you find out that maybe the microscopy was not really adequate or something was wrong with the sample preparation. So the take-home message here is that this is really an iterative process. Yeah, so typically really in the image analysis, you find out how to optimize the microscopy. So main recommendation here is to start such projects with a very minimal data set, not acquire thousands of images and only then think about the analysis, because the risk is high that you just cannot do it. So what's the aim of this analysis? Well, one thing is to quantify, reproduce, and also get some statistics. So for example, here, you could just publish these two images, but much better is to quantify them and then get a plot like for example, here, the fraction of the cells that are not in interface. And you would also like to have some statistical significance here and then reproduce this over many data sets. Another typical task in bioimage analysis are actually doing real measurements. So here, for example, the green dots would be kinesin motors walking on microtubules, and then you want to get actual numbers like micrometer per second in velocities. Yeah? So not just statistics, but real measurements. Now, to extract such information automatically from images, we have to know what we deal with, and typically it's sort of digital images. And they are just n-dimensional arrays. And the elements of these arrays are called pixels or voxels. And each of these pixels or voxels has a value and also indices by which you can map where it is in the image. Another important feature of images, they have a data type. I will come back to that later. That basically tells you how big of a number you can represent. Then to actually look at images, another important concept is so-called lookup tables. I have here one on the right, a grayscale lookup table, in this case for an 8-bit unsigned integer image, which the values go from 0 to 255. So we map these values onto some color, and that's how we can display it on the screen. That is nicer to perceive for our visual system. Now, very important is to consider actually the process of image formation. So you have to really know what do these intensity values in your image mean. And there are two things that happen in microscopy. One is blur. So for example, here, this you see a cell image from the bottom. And the width here of this is actually a receptor protein of this peak here at the edge of the cell is probably nothing biological. This is just the width of the light, sort of, or depending on your microscope system. So this is important to realize that this is nothing you should really measure because it's not biology. The other thing is all these little fluctuations here inside of the cell. Another question, are they real or not? Could be just noise. So photon noise, for example, or it could be really the distribution of the receptor. Again, something you have to consider when interpreting such data. The blur thing this actually can be sometimes really tricky. And the problem is that in usual microscopes, the point spread function where you acquire the data is anisotropic. And as I depicted it here, it's typically elongated along the z-axis. So if we look at this cell, and I tried to just draw the cell membrane, you see that here on the left edge of the cell, there's more cell membrane that fits into your detection volume than here at the bottom. So in fact, that the signal is brighter at the cell edge, yeah, that could be very interesting biology at first glance, but maybe it's just sort of an artifact of how the geometry of your object is situated with respect to the point spread function of your microscope. So this is very, very important to always draw these little sketches like I did and think about what do these signals really mean. Otherwise, you run a great risk of misinterpreting this data. Okay, so one discipline or terminology I want to introduce is called image restoration. So this is people and algorithms that try to actually counteract the blur and noise processes that you inevitably have during acquisition. And then you have, for example, algorithms for denoising and deblurring, and then your images can look much better. And there's classical methods like deconvolution, but now also this field actually there's a lot of deep learning methods, and I put one publication here at the bottom. So while this is very good, right, and it looks beautiful, it's in general very tricky to convince yourself that the lower image is really the reality. So there I also don't have an easy answer. I just want to point out that you should stay always skeptical if you use such methods. Okay, then another big topic, of course, is image segmentation. So that means we have an image on the left, which are just gray values, and then very often we want to count or describe the objects in there. And that's called image segmentation. And here I have a very basic segmentation workflow. So on the left, we have our grayscale intensity image. And then a very typical thing you do, for example, for fluorescence microscopy data is you apply a so-called threshold that says all the values that are brighter than a certain value, and in this case, I think it was the value 73, they are above the threshold, so they get the value 1 in this new image, and all the values below I put to 0. This is also called a semantic segmentation. So, and in this case, a very simple one, just a 2-class semantic segmentation. I segment my image into foreground pixels and background pixels. And then actually what I have is a so-called binary image, or also called binary mask. Then very often the next step, it's very important, is called connected component labeling. So there you run exactly the connected component labeling algorithm across your image. Across the binary image to do a so-called instance segmentation. And then what you get is a so-called label mask image, typically of an unsigned integer data type. And there, all the pixels that belong to object number 2 have the number 2, and all the pixels that belong to object number 1 have the value 1. And this is a very standard way of representing object segmentations. So if you have such a label mask image, then you can actually start measuring so-called object features. On the left here, I have just an example of 2 shape features. So for example, you could just measure the area of each object in number of pixels, and then you can find out what's here the biggest cell. Or for example, you could measure how round they are. So there is the circularity feature where you divide the area divided by perimeter squared, and the closer this is to 1, the rounder it is. So there you would find out, for example, that this is the least round object. You can also measure intensity features. So for this, it's not enough to only have the label mask, but there you actually combine the label mask with the intensity image, and then compute inside each of the labels certain intensity features. Here, for example, I have the maximum gray value. You can see, for example, here, this is in fact DNA. The DNA was most condensed in the upper left cell. That's why we have the highest maximum value. What's very interesting here is, in fact, that the sum intensity is the same for all the free cells, which sort of makes sense because the DNA doesn't necessarily change so much in content, for example, during cell division as we have here, but more in the distribution. So you can see different intensity features that can tell you very different biological readouts. That's also very important to keep in mind. Now, you can go from these sort of raw object measurements to calibrated object measurements. So for the shape features, this is done by knowing how big one pixel is in your image, and this you typically get as so-called image metadata during acquisition. And if you have this spatial calibration, you could actually measure the area in micrometers squared and not just in pixels. It's interesting to note that certain features like circularity are not affected by this. Yeah, it doesn't matter for an object how round it is if you measure this in pixels or in micrometers. So these are things that are not affected by calibration. You could even calibrate the intensity. It's not so often done. And again, there is a link of a paper where they do this, so-called fluorescence correlation spectroscopy-based intensity calibration. And this is very nice if you manage this, because then you can actually go from just gray values to, for example, number of molecules in a certain place, or the concentration, like how many do you have per micrometer cube. So this is a bit advanced, but if you can get that working, it's of course great. Okay, then image processing. That's a term that's, I would say, typically used. It's like image in, image out. And in the middle, you do something, and this can be called an algorithm or a filter or an image transform. One very prominent example are so-called convolution filters. So here on the left, we have a relatively noisy grayscale image, and then we do an average filter where we then smooth the image and get the values on the right. So technically, how such an average filter is implemented, and also the nomenclature is, for example, here it was a 5x5 convolution kernel, where each element, and I didn't draw all of them, but there are 25 of them, has a weight of 1/25. So the algorithm is you multiply each of the pixels in this neighborhood of the central pixels by 1/25, and you add up all of them, and then you get the average value, in fact. And that's how you get the new image. One important detail here is that this is in fact wrong. What's here on the right, this is not the value that you get. What you really get are these values. And that depends now what data type you choose for your image output. If it's just like the input image, an integer image, like you might have gotten it from the microscope, it just cannot represent all these floating point values. So depending whether you care now about all these decimal points or not, you have to change the data type of the image when doing these filters. So these things in practice are choices that you typically have to make and shouldn't forget. Just because these convolution filters are so important, here's another example, a 3x3 kernel. Now it looks a bit more interesting. We multiply all the values in the left by +1, on the right by -1, and in the middle by 0. So the formula for the central pixel would be actually depicted here. And this is, for example, a very simple edge filter. Yeah? So this is how you can enhance edges in image. And you can invent really many kernels. So there you can get very creative, and there exists infinite number almost of them. So why do we actually do image processing? The main reason I would say is to facilitate image segmentation. So for example, if we have the image on the top left and we apply a threshold, probably the best we can get is this on the right, not really what we want. But if we first apply an average filter and then threshold, we have a nice semantic segmentation here. So image processing and filtering is really quite often just to make image segmentation possible. And here I want to now explain one a little bit more complicated filter sequence, in fact. But it's one that's very useful in fluorescence microscopy. the so-called morphological filter, so-called top hat filter. So on the left, we have a cell and we have all these vesicles, and we would like maybe to count them. Yeah. So if we try to threshold the cell on the left, we see that we get nowhere. And the problem is that the intensity in the nucleus is actually higher than the vesicles in the cytoplasm. So we somehow have to get rid of all this background. Now the top hat filter does that in the following way. So the image one to the right, we do a so-called erosion with a certain radius, and there the algorithm is we replace each pixel in the image by the minimum value that we find in a certain neighborhood, and that effectively gets rid of all the bright dots, right? Because you always can find something dimmer in the neighborhood. But what also happened now is the nucleus was shrinking. Yeah? At the edge of the nucleus, you replace all the pixels by the cytoplasm, and that's actually something we don't want. So to restore that, then we, in fact, dilate again the eroded image by the same radius, or in other words, we do the maximum of the minimum, and that whole thing has a name because you use it so often. It's the so-called opening of the image, and then we have actually a quite good approximation of the background in the image without the bright dots. And then the final step would be we subtract from the original image the opening of itself. And this is the so-called top hat filter, quite famous filter. And then we have only the dots, and now we can easily threshold it and count it. So this would be an example of a bit more advanced filtering sequence. So again, a very basic job of a bioimage analyst is you try to process an image such that you can threshold it to get the segmentation. Now, this task can become extremely difficult. For example, here, you may want to segment the mitochondria, this other part, the cytoplasm, the endoplasmatic reticulum in this electron microscopy image. And you have to find all the filters and thresholds that do that, and this actually can be really, really tough. But luckily, these days, we have powerful machine learning methods for this. So they work that you just annotate here on the left a few example pixels, and in fact, then they try to find for you all the filters and thresholds that would give you the right, in this case, semantic segmentation. And there's now a distinction between like classical, almost now shallow learning, and the little bit newer now deep learning. And the distinction is that in the shallow learning, typically you actually give the filters that you want to apply, and you also only apply one round of filters, and then the algorithm is just learning all the thresholds. With deep learning, you typically apply a sequence of filters, so filters of filters of filters of filters, and the more you do this, the deeper the deep learning is. And the other important thing is you actually don't specify which filters you give, but these are all convolution filters, like the kernels I showed you before, but the entries there, yeah, like the 1/25 or plus and minus 1, you don't give them. They are also learned by the algorithm. So the algorithm learns also what is actually the best sequence of convolution filters to solve the task. And that's the deep learning methods. All right, so if you have done all of this, quite often the output of such a bioimage analysis task is you have a lot of images in the beginning, and then you have a lot of tables in the end. So what's often a bit underestimated is you have to check that these numbers are correct, right? If you automatically process hundreds of images, there's for sure somewhere a bug in your code. So you have to somehow have a strategy to go back from the table to the images and actually find software that ideally allows you to click somewhere in the table and then you would see the image. Or even better in the plot. So for example, here I have some real data where we quantified Golgi fragmentation with different treatments, and we clearly see, yeah, the no-codazole treatment does the biggest fragmentation. But is this true? Yeah. So, and each dot here is one cell. So what you really want is you want to click on the dot, and then the software open for you the corresponding image and tell you also which cell that was. And in fact, in these here, this is a cell that has a lot of fragmentation. And similarly, if you click on the control cell, you also want to convince yourself that here this works. So actually, it's really worth spending a lot of time also generating these images, right? So like you have the intensity image overlaid with clearly nice outlines of the segmentations and so on. So I spend usually a lot of time trying to produce high quality images that are easy to inspect later. Yeah? And also we spend quite some time developing software where you have these sort of plots and you can click and you can see the images. So this is really important in practice, otherwise you just don't know what you're doing. Another thing you should do is you should also then report in your publication how well your automated algorithm actually works. And for this, one should typically manually segment a couple of cells and then compute parameters like here the precision and recall. So basically compute ratios of true positives, false positives, and false negatives to quantify how well your algorithm is performing. So that's something one should do. Now, something about image data format. So this is really like the bread and butter, right? So we need to somehow open and save images. And unfortunately, and I think, for example, in gene sequencing, I think there is this FASTA format. There is a standard, unfortunately, with image data, and I think it's also because it's still a younger field, bioimage informatics. We suffer here really a lot from more than 150 formats, and every microscope manufacturer makes a new format. What keeps us sort of still hanging on is the heroic effort of the OME consortium. They maintain this bioformats library, which allows you to open all of these formats, but it's really not a sustainable solution. So we really need a community standard format, and ideally now with all this cloud computing, a cloud compatible format, and there's light on the horizon, I would say. Good news. So the very good candidate for this is called OME-ZAR, and there is a GitHub repository where this is discussed, and also on the forum that I will discuss in a minute, you can follow with this text. So if you are a bit more of a techie person that cares about image data formats, please get involved here. It's, I think, an extremely important effort. Okay, then I want to do something a little bit less theoretical and talk about some actual platforms where you could do image analysis, and that's a really tough job because I have to pick something now, and there are so many. So I made the choice to say, okay, I present 4 of them, and the criterion was they have to have more than 1,000 citations, and I should personally know a couple of people at least that use them, and I apologize for all the tools that I don't manage now. Again, this is a tough choice. So an easy one with, I think, more than 10,000 citations is Fiji. I think basically everybody has it somewhere on their computer. This is a software that comes with a graphical user interface. It has really many, many plugins that makes it very powerful. The other good thing is now there's a relatively new core library called ImageLib2, which enables processing and visualization of terabyte-sized data sets. And this is basically so big data viewer ecosystem within Fiji. Another really powerful feature is the script recording. So you can click around in the GUI and actually record what you did into a script. So this is also, I think, one of the key features. And there are several languages. So for beginners, the ImageJ macro language is very, very good. But then you can also get a bit more advanced and program in Groovy or Jython more directly on the Java API. Another software also with more than 10,000 citations, Cell Profiler, and I use this personally really a lot. This is purely graphical user interface, so no scripting, and it's really great to design image analysis workflows. In fact, that's the only thing you can do. So you see here on the left, you have to kind of actually immediately specify a workflow of steps that you do, and it's and then for each step it actually shows you for the current parameter settings what would be the result, and then it's very convenient to just change like a threshold and then check just for one step how the output would have changed. So it's really fantastic software. I can highly recommend it. And it's inbuilt batch processing. So it's already done that this will then run on 1,000 images. You have to do nothing extra. So highly recommend it. Then if you're a bit more into scripting, another ecosystem, I would say, and in fact, Cell Profiler itself is written probably with this in the back, is called Scikit image. So this is a Python library. There you really have to write code. It has a lot of functionality that plays well together. So it's a consistent ecosystem. I just checked. It has really, really great documentation. And the really good thing with this is that now most of the, I mean, in Python, you just find so many things. For example, all the deep learning now is mainly Python. And then you can easily just chain together your Scikit image script with some deep learning. And the other really good news is that now, since maybe already a couple of years, there is now also a really good viewer for images in Python. This was always a problem, that it was hard to look at images in Python and people would then open Fiji. But now you can directly from Python launch this Napari viewer, which is really great. And to look at images as convenient, or maybe depends, maybe find it even more convenient than Fiji. And you could even inside have a script editor where you can write your code. So this Scikit image Napari ecosystem also highly recommended if you like to write code. Then there's Elastic, actually currently, or not currently, so developed at EMBL, my colleagues. This is a really good software if you want to do machine learning. So it has great, it's graphical user interface. It has great functionality for all the annotations. So you can label different parts of your image and then get immediate feedback of what the machine learning algorithm would give you as a segmentation output. It started with the shallow learning algorithms, but now, since quite recently, actually, they also support deep learning. And there's a very interesting initiative. You should check it out, the so-called bioimage model zoo. So this is a great effort to standardize all these deep learning models such that they can be just run in different platforms. And for example, Elastic, but in fact also in Fiji, there is the Deep Image J plugin, which also aims to be able to run all these deep learning models. So very interesting. Highly recommend it again. Have a look. Then also more than 1,000 public citations. QPath, a bit more specialized. So it was really made for digital pathology, and there the thing is, first of all, these images look a bit different typically. They have more this H&E stain, color camera sort of images, not so much fluorescence. And they have also this really large 2D slide scanner images that are actually hard to open even in Fiji. So it can handle those very well. It has great tools for segmentation, and it's very good for annotation. So many people just use it now for that, even if they don't do histology. And you can also do scripting there, in this case, in the Groovy language. So this is also a Java-based platform. All right, so some more practical tips. If you want to learn more, and for sure you want to learn more because I could only talk about so little today, there is something I would like to advertise here. Is if you Google Neubayer's Academy, so there is a lot of nice webinars like the one today about all kind of aspects of image analysis. And for example, about QPath, about Cell Profiler, you will find a whole hour about each of them. So the Neubayer's Academy, Google that and have a look. Great stuff. Then you can go on the forum. Actually, there is a thread I launched at some point, which is just where people just put links to recommended reading and viewing for bioimage analysis. So there you can also find links to a lot of courses and material. The other thing I think you should just check, maybe there's a professional bioimage analyst working at your institute already, so get in touch with them. So of course, a good idea. And then finally, since I do have time, I want to show the forum here a little bit in more detail. So that could be a place, even, I don't know, if your level is you don't know anything about image analysis, or maybe you know something but just want to check if there's an even better algorithm or something, the forum is the place. So I post also a lot there and ask people, hey, would you do it differently, or what would you do, and so on. So I will just live demo this now, so that you can see how you could do this. So this is the forum, and you see there is tons of softwares that committed to use this as their main forum. There's Cell Profiler, there's Fiji, there's ImageLib2, there is QPath, I can't see it now, and so yeah, OME is there, so basically everybody's on board, which is great. So you reach all of them if you post something there. So if you have a question, you can click on New Topic and then type, for example, help with nuclear measurements or something. And then the category here would be you need help with image analysis, and then you have to put at least one tag. I don't know, you could put segmentation. And then you have to type something here, and I prepared a little template. And so what you have to keep in mind, the people that answer there, right, they are not paid to help you, okay? So you somehow have to be nice and make it attractive and easy for the people, yeah? Otherwise nobody will. So for example, you could write something like this, right? Dear bioimage analyst, may I kindly ask, I have data shown like in the two images below. And then what you should do is, and then figure out here the syntax, provide download links to the raw data for a control and a treatment, ideally, and then say what you want to do. Yeah, I would like to automatically measure that the treated cells are larger. Would you have some advice for how to approach this? And then you could also write, if you're already familiar in Python or something else, that people then could answer you with something that you can actually do. And what one can also do here, and I would recommend that, is you can in fact just copy and paste the example images. So that's also important to make it visually attractive, because if people see a post with just text, usually probably go to the next topic. But if they see, oh, you want to measure the difference between these and these cells, and you provide the download links to the raw data, some people might just post the complete solution for you if you're lucky. Yeah. So you just have to basically make it nice and attractive for people to answer. But then otherwise, it's really a great place. Okay. That was it. So I would like to thank a lot my colleagues at the Advanced Light Microscopy facility and also at Electron Microscopy core facility at EMBL, the Peppercork, Ellenberg, Schwab, and Kreschuk labs at EMBL, Antonio Politi, not any longer at EMBL, for developing a lot of teaching materials, Kota Miura, really amazing. He founded this network of European bioimage analysts together with some people, so that's an amazing task. And I would like to thank all the people that hang out with me on the forum. And I would like to thank you, and then I'm happy to answer any questions that you might have.
Thank you, Christian. Before we move to questions, could you go to the next slide briefly? So I just would like to make you aware of our upcoming webinars in Methods in Bioinformatics series. So the first one will be in Methods in Genomic Variant Calling, and it will happen on the 3rd of March. So have a look at our website and training and then webinars to have a look at this series. So the next slide, please, Christian. So you can also find recorded webinars like this one and any other webinars in our on-demand training. So I do provide the link here, but if you go to on-demand in training in recorded webinars, you will find a list of webinars. And please let us know your thoughts at the end of this webinar by answering the feedback survey. So yeah, now we can move to the questions. Next one, please. I think it's just a question slide, Christian, the next one. So I see in the Q&A, we have 5 questions. So
okay. Yeah.
Yes, can you see them?
Yeah, I can start with the top voted one from Fernanda. So for co-localization, would you know and recommend a plugin in Fiji? That's a very interesting question. So I would recommend to watch the Neubayer's Academy talk about this. It's from Fabrice Cordelieres. So he is the author of actually one of the co-localization plugins in Fiji. I think it's called Jacob. And he also wrote 2 papers about this topic. So I would definitely check them out. And actually, it's interesting if you listen to his talk, his recommendation in the end is you should probably write code for your own method of co-localization. Yeah, and he's the author of the plugin. So, and I agree with him. Co-localization is very, very difficult, and for each problem you have to just running some standard code will probably not work. So you have to think about it, probably invent your own method, and there I would really highly seek some professional help from someone that is really into bioimage analysis. It's very easy to, it's just really difficult, difficult problem. So I would not just use some plugin, would be my answer. Okay, who has the next most votes? Susan has a lot. What strategy do you recommend to segment image based on the color intensity? Yeah, interesting. So there I have not much personal experience. I would actually probably look into QPath because they, for this, what I just said, have this kind of data, yeah, that it's from histology, and then you actually do segmentation based on the color. So I would look into QPath and maybe again look at the Neubayer's Academy QPath talk and then see if you can solve it with QPath. I think they might have ready to use things for that. Okay, then Rajini, sorry for the name. Hope it's okay. Which platform would you recommend to use for 3D image analysis? Very good question. So in Fiji, I mean, Python programming for sure. In actually Fiji, so for teaching now, we always use a plugin called, or a lot, not always, MorphoLibJ or the 3D image suite. So there are 2 very good Fiji plugins for 3D analysis. Also in Cell Profiler, actually, if you download the latest version, you can do 3D. The problem there is a little bit that the visualization in 3D is not so good. So it's a bit hard to inspect the images at the moment, but you still can do it. Otherwise, we actually also use commercial software for this if it's big 3D data, because 3D visualization is not so easy. So we use commercial softwares like Imaris or something like that. So I think that would be my answers. Then Victor, what is the most versatile image format? By versatile, I mean the format that tends to be supported by most machine learning methods. Yeah. If I would know the answer, that's why I was saying we should really find a solution there. That's why I'm very much active with this OME-SAR, because I hope that we will finally have a format that will answer your question. I think for now, the best standard might be OME-TIFF. So I would say that's maybe my answer. Then here, Niels. Okay. Are there agreed upon thresholds when an object segmentation should be considered a true positive, or is it recommended to show precision and recall over a range? I think the second. So there's just no standard way of setting a threshold. I think you have to do the precision and recall. Yes. Then Sandra, EZ co-localization is also interesting to check out. Good, fair enough. Yeah, so I don't know it, and again, I would just be very cautious. I mean, it's okay, but yeah, thank you, Sandra, for mentioning that one. Might be very good. Then we have Eleftherios. Are there any tools available for segmenting nuclei from spatial transcriptometry images using R Studio? Yes. So in R, there is a package called EB-Image. I haven't used it, but I think you can do pretty serious image processing with this also in R. So this is all I know. So check Google for EB-Image. I think that's the package that I know. Maybe there's something newer, but that's the latest one I know. Then,
now we have Susanna.
Wow, yeah, you have 10 votes.
Yes.
Sorry. Yes. Do you organize or aware of any practical course for PhD students, postdocs, 1 to 2 weeks to improve image analysis skills this summer? So there is, I don't know if they do it this year. So you have to Google for CDAS. So Z-I-D-A-S. They have a summer school 1 week every year. Other than that, to be honest, nothing comes to my mind right now for longer schools. So the Neubias community, we had every year, I think twice even, training schools, but the funding ran out. It was EU funded. So this is now on hold. We are trying to get funding again, and then the Neubias kind of community would be another place to look, but at the moment it's unfortunately paused. Yeah. Okay, so sorry, Susanna, not too much that I can offer there right now. Then Akansha, sorry. I want to measure the intensity of an image over a period of time. May I know, instead of individually marking voice in each image, is there another way? Yeah, so of an image, you mean maybe objects in an image or the whole image? So generally, the thing to do is this label mask image concept. Yeah, so you have to somehow create a second image that's a label mask. So which I explained in the talk, and then like MorphoLibJ or 3D image suite if you use Fiji, or also Scikit image in Python. I mean, this is really the standard way. So you somehow segment your image and you get a label mask image. And that's basically then sometimes can be difficult, or you can use machine learning, but you need a label mask image. And then you combine the label mask image with the intensity image for each time point, and then you can automatically measure things. That's always sort of the way to do it automated. Okay, Ramakrishna, what is a good method of documenting image analysis pipeline? Yes, so basically the best way is you just write code. So if it's a script that you run, then this is the best documentation. So if you use Fiji, then actually macro recording yourself when you click around is the best way to document it. If you use a software that only has a graphical user interface and you cannot produce a script, then I think we discussed this. I think maybe the best you can do is you can make a video of yourself using the software. Unfortunately, it's then the best way of documenting it. So, but the best way to have a script that can be run in some programming language. Okay, then Carol, you haven't mentioned MATLAB. Why is that? Yes, because maybe I didn't put it in the title of my selection. I then decided I want to restrict myself to open source. And MATLAB is commercial and it's not, you have to pay for it. Not everybody can have it. But otherwise, MATLAB is great. Yeah. So it has a lot of things. It's just, a bit harder to share because not everybody might have it. I think these days, in Python, for example, you can probably do most of it, but no, it's a fantastic software. It's just that it's commercial, and I wanted to restrict myself to open source freeware. but nothing wrong with MATLAB. And there's a lot of things for image analysis. Okay, Josefina, I would like to know a good method for determining the right radius to apply in filters so as to avoid creating more problems instead of solving them. Yeah, again, no easy answer. I mean, that's also the visual thing with images. I mean, you just have to, okay, let me give a different answer. So probably the best way would be if you were painting a ground truth image, yeah? So you make a manual segmentation first of the results that you want to have. And maybe as a label mask image. And then you have a way to automatically compare for different filter radii, the label mask image you get from your algorithm with your manual ground truth. And then you could automatically almost select the filters. And then you are in fact already very close to machine learning. That's basically what they do for you in a more black box way. Yeah? So I don't know if that was your question, but yeah, otherwise it's just trying different radii, unfortunately. Drusha, could you suggest some good methods for processing images with outer fluorescence? Phew. That's why I had the slide in the beginning. The best is, of course, to work on the sample preparation and don't have autofluorescence. I mean, for sure, you have to take always images, like control images, that you're really sure what is fluorescence and what is autofluorescence. And then maybe you can find something that allows you in the final image to automatically distinguish it. The other answer is if your autofluorescence is more of a sort of spread out background and maybe your signal is more like detailed peaks, then things like this top hat filter that I was showing are powerful in removing sort of blurry background, but that only works if there's a so-called separation of spatial scales. If the autofluorescence has the same, let's say, granularity than your signal, I don't know, then it's just tough. And I think we would have to look at it in detail, but it can be very difficult and close to impossible. So no easy answer. Yeah. That's difficult. Okay, let me scroll a bit. Thank you, by the way, for all these great questions. It's really good. Okay, Animesh, which deep learning library, PyTorch, TensorFlow is recommended for doing things like image clustering, cell counting, and so on? So I'm not the super expert here, but I'm also not sure if this is really typically done with deep learning. So I think there is in Python this whole, I think it's called sklearn, or is it scikit-learn? I don't know, you will find it. So there is like stuff for general machine learning, not necessarily deep learning. I think clustering, for example, I don't know. Otherwise, in terms of whether to use PyTorch or TensorFlow, I think you're pretty much 50-50 from the people that I know. So I cannot make a strong recommendation there. And again, if this was not a good answer, ask on the forum, okay? Just anyway, always, you might find people that are more experts. Okay, now it's tough because there are several with one. Can I see who was waiting longer? Yes, I can. Okay, so then I try to go to the oldest one. Oh, cool. Thank you very much. Yeah, thank you. Which softwares would you recommend for analysis of macro images? Oh, so I haven't worked with this, and I don't think that qualifies for a specific. I think all of these platforms could work. If the images are really, really big, and also I think it looks like you might have a color camera image, maybe QPath. Maybe QPath. Maybe QPath is your thing. I'm not sure. Try QPath. Joseph, are there certification courses in bioinformatics? You mean like maybe probably bioimage informatics? I'm not nothing. I don't think of anything that has a, like, say, EU standard or something like that. Probably maybe at your university, but yeah, I basically I don't know. Fernanda. Okay, it would be nice that Neuweyers can offer a practical course online. Yeah, that's a good. I mean, you can watch all these videos, right? They are sort of, but then they are not without feedback. But that's no, that's definitely. And yeah, if we can find funding again and enough people motivated, I think online courses would be probably also on the list. Yeah, thank you for the suggestion. Josefina, my lab has an old Emaris license. This is good software, or should I keep learning Fiji? No, Emaris is great. It's just, well, it's just for publishing, I think, for example, right? It's always nice if it's something that let's say the reviewer of your paper, that they can reproduce, and they will for sure have Fiji and be able to run a macro, but they might not have Emaris. Yeah, so that's always, so in case of doubt, I would probably say use something that potential readers of your science can reproduce, but otherwise Emaris, as I said, we also use it, and I think it's fantastic. Yeah. So I wouldn't throw away the old license. Then Chini, thank you for, yes, thank you. Like you said, I have been using MorphoLibJ, good choice, great stuff. However, there's no tool for deselecting. Cells are close to each other, the program. Yes, there is. So if things are touching, the sort of algorithms that you should check out are called Watershed Algorithms. That's basically sort of the tool, the algorithms of choice for dealing with touching objects. And then you should look if like the cells, if they touch, if it's dimmer where they touch, then you can use this information to draw the sort of dividing line there. If they have the same intensity, then what you have to do, sorry, the technical, you have to first do a so-called distance transform on the binary image, and then you sort of split them by shape. Actually, inside Cell Profiler, this is also very nicely implemented. So you might want to have a look there. There's a split touching objects option in Cell Profiler. So this should be somehow possible, and the watershed is the sort of thing you have to look into. Could you please repeat the name of the website? Yes, so I can I type somewhere? Ah, yeah, type. No,
yes.
Yes, so they used to do this every year. I typed it. Hope you can see it. Then I maybe go now to the old ones. Is there any specific software for?
I will now sort for most recent, so you can go to the old ones.
Old, yes. Okay.
Yes, if you, yeah.
SEM, so with SEM you probably mean like serial electron microscopy or something, specific software or method. Yes. So there is, I think MIB is the name. I think it's called Microscopy. I'm really, really sorry if this is now. So this MIB, I think this is really made for electron microscopy, free software. So I think that was the name. Check it. Otherwise, ask again on the forum. That one is really specific for EM, I think. In terms of colocalization, is using AND image calculator. Yeah, the AND. Yes, good. Yeah, so for example, if you have two fluorescence channels and you have segmented both of them, then you can count like the number, like you do a semantic segmentation, foreground, background in both, okay? And then you have two binary images. And then indeed, if you do an AND operation, you would get all the pixels that you have segmented in both. And then you can, for example, divide the number of pixels in the AND operated image by one of the two others. And that's actually one way how you could measure co-localization, yes. So that's one of the most standard ways indeed. Marion, our studio, yeah, I said already, EB image, I think, so that's the one I know. I can type it also. I think that's the thing in R. Joseph just said or, maybe as an answer to the and. could be, sorry, so I'm not, maybe I was wrong, but I think and, yeah, or can, yeah, probably, as I said, for co-localization, the best is to invent your own method, yeah. My question has not been answered, Okun. Sorry, which question? Can you repeat it, maybe?
Yeah, let me check here.
Or maybe we kicked it away.
Yeah, I can't see. Could you write again?
Yeah, sorry. Okun.
Oh, here, I see, maybe it's that one. Which softwares would you recommend for analysis of micro images? Say those obtained from bacterial culture plate.
Ah, macro. Yes. No. Sorry. I think I would start with QPath maybe. That's because it sounds like it looks a bit similar to histology images. So I can type. I would start looking into QPath maybe. But then Kundi, do you accept any bioimaging job shadowing trainee? Not really officially at the moment, but you can still send me an email and we can discuss. But there's nothing, no official program. Yeah. Okay, Risa, which softwares or plugins in Fiji would you recommend to use for segmenting, tracking, moving objects in videos? Yeah, so if it's round, okay, so in Fiji, there for tracking, I think there is a kind of easy answer. There is TrackMate. And if your objects are sort of dots, then you can also use TrackMate for segmentation. If they have a shape, then actually you would like to do the segmentation outside of TrackMate. So then the workflow I would probably do, because I like MorphoLibJ, I would use MorphoLibJ for segmentation. Then I would produce a label mask image, or not, but as a function of time. And I would go with this into TrackMate. And fortunately, since very recently, this is possible. So you can actually, as an input for tracking, use label mask images in TrackMate. So that would be if you want to segment outside TrackMate. But otherwise, TrackMate sort of for tracking, yeah. Okay. There's someone with two. I lost connection, so I don't know if you already talked about it, but what do you think about using ImageJ? It's good. ImageJ is like always what I would start with, so no, definitely. For outer fluorescence, I would say the best is to use the right filters in the microscope and choose the right fluorophores, or maybe use spectral detectors. Yeah, so this was Fernanda answering to previous questions. I fully agree. I think I also said that the best way to deal with outer fluorescence is on the microscope and not during image analysis. It might be close to impossible. Fernanda, yes, I totally agree. Then, when I repeat an experiment and have different background noise in different repeats, is it possible to automatically yet dynamically adapt the thresholds? Yes. So there is many automated thresholding algorithms. Also in Fiji, actually, there's a very nice, I can type it. So there is a just, I think it's called auto thresholds, and they have a very nice try all function. So you can just input your image and see. And there it's important indeed to check, to find something that works for all your images. So these automated thresholding algorithms, they all have their little regimes where they work more or less well, but they definitely exist and exactly for your use case, and it's fair enough to use them. So yes. Okay, final question. Tabitha, what plugin program do you recommend to register slices for 3D images? Okay, so actually, this is funny because so in Fiji, there is something I just recently learned, which is really nice. It's called Shift Align Stack or something. So this would be the Fiji thing. If you really get more serious into it, okay, if you have electron microscopy, there is a whole thing called TrackEM in Fiji. If you really want to get into registration, I use a lot something else. It's called Elastics. I write it. This is outside of this whole. It's a different ecosystem actually, but they distribute a binary. It's fantastic. It's coming more from medical, but it's really great for registration. So